So let's get started. It's a pleasure to introduce our next speaker, Bernhard Lippert, who was a postdoctoral fellow and adjunct assistant professor with Barney Rosenberg in mid-1970s. So during his time at MSU, he worked on the hydrolysis products of cisplatin and other novel platinum pyrimidine complexes. And after returning to Germany, Dr. Lippert served as a professor of chemistry in the Technical University of Dortmund from 1988 until 2011, I think, right. Until his retirement. So he's gonna talk to us about working with Barney Rosenberg on the platinum pyrimidine blue complexes.
Thank you very much for your kind introduction. It's a great pleasure for me to be here at MSU and to tell you about a story that was started by Barney Rosenberg and his co-workers and which has fascinated me ever since and in fact still fascinates me. Before starting, I would like to thank a number of people and institutions. And in particular, I would like to thank Barney and Rita for their enormous hospitality during my stay at Michigan State and that of my family. The work I'm going to present to you today largely developed after I had spent the time at East Lansing. But since Barney put me on this track, I think this should be, he deserves this honor. This is a picture from Barney's group, or part of his group in the 1970s. At that mid-70s, we were 4 postdocs, chemistry postdocs. So this was Ron Pollack, Jim Bernhard, Paul Schwartz, and myself. And of course, you recognize Loretta Van Camp. And this work, they worked on platinum blues and were directly involved. Of course, this rested on earlier work by people which I have mentioned here, some of which are here in the audience. This is a nice picture of Barney, which I received from Tina many years ago, and that's the famous license plate of the Oldsmobile of Barney. Now, Barney was a great mentor for me. He was very inspiring and generous in all ways. He was no whipmaster at all, even so he's sometimes ridiculed with this expression. He allowed me a high degree of freedom in designing experiments and also in determining working hours. The group I joined then was very new for me. It was interdisciplinary. I came from a purely inorganic chemistry group, and that gave me a lot of insight into the chemistry and biochemistry of these platinum drugs. We had also a good time, remember playing basketball or even soccer during lunch break. My projects then were essentially 2, the hydrolysis products of cisplatin, their isolation and characterization, and their structural analysis. And the other big theme was the platinum pyrimidine nucleobase complexes discovered in Rosenberg's group. I also did, by accident, some organometallic chemistry, which was concluded or finished only later when I was back in Germany and when we really got several X-ray structures of these compounds. Coming back to the platinum blues, they were developed, as I said before, in the Rosenberg group, mainly by Sami Mansi, who is also in the audience. He found that reacting polyuridine with this diaqua species, it developed an unexpected and unusual blue color. Later on, it was found that you get blues also with uracil, uridine, thymine, and related ligands. In 75, the first report on the anti-tumor activity of such compounds emerged. There was always a problem with reproducibility of these compounds, and so it was eventually agreed to have a standardized procedure to make such blues, which is listed here at the bottom. And I'd like to point out just one important detail, and that's the 1-to-1 ratio between the ligand and the diaqua species. If you look up the internet and search for platinum blues, you may come up with this page. It has nothing to do with chemistry, I tell you. It's a sad story from pop music where somebody thought he was cheated to get a platinum record. But I always wondered whether this author may have been close to chemistry at one stage to come up with this title. This is, in my opinion, a very crucial slide, which you should keep in mind to understand what I'm going to tell. So when you react the diaqua species, which has 2 open sites for reaction with any ligand, for example, with a pyrimidine nucleobase, you, in a 1-to-1 ratio, you cannot expect to only form the 1-to-1 complex. Depending on relative stability is constant, you will also always also form the bis ligand complex. And for every bis ligand complex that is formed, you will, for every molecule of such a compound, you have a free molecule of the diaqua species. And here the problem starts. First of all, if you take the 1-to-1 complexes, it can self-condensate either via the pyrimidine ligand in our case, or the aqua group forming a hydroxyl-bridged species. And they can react in addition with the free diaqua species to give blue compounds. And you can expect to get different blues. And today, only one, which I'm going to show, is really X-ray structurally characterized. And that's if you start from a so-called head-head dimer reacted with diaqua species, and then you get a dinuclear complex, which eventually forms a blue that has been crystallized by Steve Lippert. Here are the, maybe if you use an excess of the pyrimidine over platinum, you will always get a bis complex, sometimes with an extra ligand in the outer coordination sphere, but you will not get a blue. Here are the ligands to show you in more detail. Those are pictures of one substituted pyrimidine basis, so to speak, pyrimidine model nucleobases. And it's the N ions, this is a uracil, this is a cytosine, that are responsible for binding metals through the N3 position, the exocyclic 4 position, an oxygen or an NH group, a deprotonated amino group in the case of cytosine. And there's also the possibility for oxygen to bind metals. As I will show you in the third part of my talk, if you have a proton here, so if you take the parent complex, the situation is even more complex. So we set out to try to make as many compounds as possible to understand the complexity of the system. And we used one trick which is mentioned here. Occasionally we use a palladium entity, say ethylene diamine palladium, which behaves very similar as cisplatin, but it reacts much faster. So this, from a structural point of view, this is very similar chemistry, but it is much faster. And we have done this also in combination with platinum binding. Occasionally, we also replace the ammonia ligands of the cisplatin entity by other amines. And I'll show you one case where we mix cytosine and uracil. Very quickly, this is a busy slide. I have to go through the platinum pyrimidine blues as prepared by Barney contain a mixture of different compounds, diamagnetic species, which are colorless or slightly yellow, and colored paramagnetic species. We call them blues, but sometimes they are purple or even red or green. And they seem to be in slow equilibrium. The colored species are cationic. Under certain conditions, as I will show you later, you can crystallize such compounds, or in fact, there is only one crystallized compound, which I'll show you in a minute. Animal testing, I mentioned this before. These blues even were in clinical trial for a short period of time. And much work in the Rosenberg group has also been done to use these blues as nucleic acid stains, either for free nucleic acids or nucleic acids inside cells. The reason is that they are positively charged. These nucleic acids are negatively charged polyanions, and of course, there is a strong attraction between. So the problem of reproducibility I already mentioned. As to the properties of these compounds, it soon became clear that the color came from charge transfer between platinum centers, and this is always the case if you have different oxidation states, not just platinum 2 as in cisplatinum, but also higher oxidation states, +3 or +4. And in order to generate such systems, you need to have at least 2 or possibly more platinum at close contacts. So if you remove a proton through oxidation, then you get an exchange of this charge over several platinum centers. And that causes the blues. We find different absorptions at different wave numbers, and the blues prepared by Rosenberg at close to neutral pH, they contain only a very small amount of platinum 3, of paramagnetic platinum 3, which I had determined when I was in Barney's group. If you use external oxidants, he used always air. If you use external oxidants, you may produce higher values of platinum 3 contents, but you have to go to low pH 0 or 1 to achieve this, and sometimes you have the problem which I'm mentioning here. Now, you see the only structurally characterized blue by Steve Lippert, that's this compound. It is prepared by starting out from a yellow diamagnetic compound, this so-called head-head dimer. Head-head means we have the 2 ligands pointing in the same direction. So if we point the 2O4 to one side and add the diaqua species, we come up with this dimer. We had done the structure as you can see here, and Steve Lippert now oxidized this at low pH, and 2 of these entities come together, and you lose one electron by the oxidant, and then you get this compound where the distances are much shorter, the intermetallic distances, and this contains formally 1 platinum 3 and 3 platinum 2, giving an average of 2.25 oxidation state per platinum. As I said, this is the only pyrimidine-based blue that is known to date. We have done a similar reaction, as I show you in a minute. We started out with this compound, just put it in water and according to some results from Japan, they think that it's this compound which is active as an anti-tumor agent. Yeah, we have done a similar reaction. We took this dimer and we just dropped it in water and allowed it to stand for a long time. So this was were very mild conditions like the Rosenberg protocol. And what we observe originally is the solution is yellow, and then it turns green, blue, and eventually dark blue. And it has a different absorption spectrum than the Lippert blue. We know the extinction coefficients of the Lippert blue, and so from our spectra we can calculate the amount of Lippert blue in a typical platinum blue at pH 6, which is completely different from low pH, and it's a very small percentage only of this platinum blue prepared by Lippert. If we follow the reaction by proton NMR spectra, we find that there is rearrangement reactions, and among others, the dimer goes back to the 2-to-1 complex and diaqua species, and they rearrange, and in fact, we found in mass spectrometry a compound which seems to be very interesting. It's listed here, has a very unusual composition, and it could be that two of these 2-to-1 complexes are cross-linked intermolecular by a diaqua species. So the dimer, the head-head dimer is just a kinetic product and not a thermodynamic product. So it forms, but if you allow it with time, it will dissociate in part again. We also found something which surprised us, but which was confirmed by others. In this reaction, we could see that slowly ammonia is released, and that's also unusual at first sight, but maybe not quite, because if you have a platinum 3, this is a D7 system, it's kinetically labile, unlike platinum 4 or platinum 2, D6 or D8, and so it is known, and in fact we have shown this in other cases, that you can easily substitute the ammonia ligands then. Suppose you have in this mixture also a platinum without any other ligands but water or hydroxyl. So you have, so to speak, naked platinum. This could also make some interesting chemistry, as shown on this slide. You see, I get used to the language some of you use today. So if you lose ammonia and generate a naked platinum, it could react in any of these cases. These are only models because this is a naked palladium. So we take the 2-to-1 complex, react it with palladium, 2 aqua species, and it binds to O4 in such a way to form a trimer, which is yellow. And if we oxidize it, then we oxidize it to a species which formally has 2 metals in +2 oxidation state and 1 metal in +3. So we have a shortening, and it is intensely blue. This is another example what could happen, but in fact, we have done the same reaction with platinum, but we were unable to crystallize it. And the same here. Here we used the head-head dimer, and we add silver ions. And the silver ions can bind to these O2 sites and then cross-link 2 of these entities, and we get a pentanuclear complex. This crystallizes the way that is shown here. It is yellow, but it goes very interesting redox chemistry as pointed out here. I switch now to cytosine complexes. Those cytosine has not been used by Barney for reasons I don't know. Maybe some of you who were in Barney's group in the early days might know, but it behaves very similar. We used it because the exocyclic amino group, which becomes deprotonated, is a better donor than O4 in uracil or thymine, and we hoped to get more information from their reactions. So we have the 1-to-1 complex, the 2-to-1 complex, and that's an isomer of this, where one of the cytosines is bonded to N4. Now, if we have the same situation, we have the possibility of self-condensation of the 1-to-1 complex through either the ligand or this aqua group, and also the possibility of condensate of reaction with diaqua species. And we have crystallized all of these compounds and have X-ray structures, but I'm going to show you just a few examples. This is a very simple one. Cisplatin reacting with 1-methylcytosine, you get this compound, and then you displace the chloride by an aqua ligand, and then you have this compound. And it dimerizes spontaneously to form a head-tail dimer. Now the two bases are oriented in different directions. Now this is an interesting compound because it is, first of all, it's chiral, and it has the ability to hydrogen bond here, as we have shown in several examples. And so this compound is interesting with regard to, say, binding to DNA in principle. From an inorganic point of view, it's interesting that we can also put metals here, and I show you one example here. We have added silver ions, and we get polymers which are chiral, and we have right and left-handed helices in this compound. And these silver ions, they cross-link in an intermolecular way, so from this dimer to this dimer, and this is just a section of one of these. Suppose you were to have a platinum, not to silver here, and instead of nitrates, the ammonia ligands, you could think of a similar structure. And in fact, if you react this head-tail dimer with the diaqua species, we get, again, a blue, but we have been unable to crystallize it yet. Just as a side point, this also forms not only dimers, but also trimers. This is now with palladium, but by Bruno Longato from Italy, there is a report that they also have platinum analogs. They bind anions very strongly. This is from cytidine, and this is perchlorate, and that's again interesting with regard to DNA. There's one other interesting reaction of this compound when we reacted with single-stranded DNA. This is DNA. I don't know if you can recognize it. It wraps around one of these trinuclear cationic molecules, and if you wait long, then this DNA is completely wrapped around several of these cations. Finally, one exciting compound which was obtained under very mild conditions, again from the 1-to-1 complex, and here you find three different condensation modes. There's hydroxyl bridging between the platinum. There's bridging which involves the cytosine ligand to this platinum, and much to our surprise, we found that also this amino group condensates with the hydroxyl group of this platinum to form an amido group. This occurs in water, and it was very surprising to us, especially also because most of the people think that the amino ligand of cisplatin is completely unreactive. Now I come back in the last part to Barney's system with the unsubstituted one. So we don't have a methyl group here or any alkyl group, but we have a proton. And that's very complicated. So the monoanions and the dianions play a major role, and even the monoanions can come in different forms. You can deprotonate it the N3 position or the N1, and there is also the possibility of tautomerism, and in fact, we have crystallized such compounds. I have referred here to a paper which was I was the only author and was the longest paper I ever published, just to show how complicated this system is. And I try to show you now a few examples. This was a key complex which we discovered in the early 90s. It's a cyclic structure. We started out with an ethylene diamine platinum bonded to a uracil through the N1 position. And this compound spontaneously cyclizes to give such a complex. We were very happy about this before we realized that this is only part of the story, because if you react uracil with the diaqua, you may not expect to get only this compound where you have this N1, N3, N1, N3, N1, N3, so this sequence, but there are also other linkage isomers possible. And I just referred to this one where we have N3, N3, N1, N1, N3, N3, and so on. I'm going to show you an example of this. These bases can also adopt different orientations, and this could be stabilized by intermolecular interactions. And to make the story short, there are many possible isomers, and we have just crystallized one of these. I'm going to show you another example. This was a uracil. Cytosine behaves pretty much the same. Unfortunately, we have not been able to crystallize any of the others, even so we think that they exist. What we have isolated is shown here. We have isolated larger system. This is a hexamer, and this is an octamer. The hexamer is still flat, and this no longer, it is folded. What we have here, we started out with a platinum and 2 cytosines, and then react this compound with ethylene diamine palladium, and then it cyclizes as shown here. And in this system, we have used uracil and cytosine, but I'm not going to discuss this in detail. Coming back to this compound, I just showed you. This is not the whole story, because here we have N1 and 3 binding, and the proton, it's a monoanion. One of the protons sits at the exocyclic group, and there is hydrogen bonding as indicated here. Now you can displace this proton, and we have reacted this compound with the diaqua species of cisplatin, and then you get this octanuclear complex, which is kind of a double decker, and which also forms a blue, by the way. So let me just show you again how the different binding ways affect the outcome of this structure. So this is the example where we have N1, the blue is nitrogen, N1 and 3 binding of cytosine. And if we add now, this is a palladium compound, and if we add palladium, we get this double decker structure, which has 8 metals. Now, I'm not going to discuss the structure for time reasons. Just show you another case where we have this N1, N1, and N3, N3 binding. So these are always the same sites, N1 or N3. And if we add platinum, we get another octanuclear complex, same composition, but it has a different structure because the exocyclic groups are arranged differently. And then you get these, you get 3 metals above each other, and this is interrupted by a single metal. So this just gives you a flavor of the complexity of this. And I'm not going to discuss the structures here. I will rather come to the end. And this is a summary of what I've discussed the last few minutes. If you think of N1 and 3 binding, you get at least different sizes, but also different arrangement, as I mentioned. And if you if you want to make larger ones, eventually this will break up, and you will be getting a linear compound. Now, let me, and this slide does not include the fact that you can bind additional metals to the exocyclic groups. So this is really complicated. Let me end with a structure which is the largest which we have been able to prepare. It contains uracil and platinum and palladium. This was the starting compound. This is your unsubstituted uracil bonded through N1 to a cisplatin. And this is the second one, and the same is here. And this green is a palladium. So we have added palladium, ethylene diamine palladium, and get this trinuclear complex. Now there is, on the next slide, there is another trinuclear complex, but I just dissected the product which we got. There is no interaction between these two halves, unless you add metal ions, the yellow ones, and then you close the circle. And the yellow palladium, ethylene diamine palladium, bind through ring nitrogen and one of these exocyclic oxygens, like in the head-tail dimers. But this is not the end, because you still have these exocyclic groups, and they can accommodate additional metals. So in this compound, we have 14 metals. This is the result of an X-ray structure, but unfortunately, if you dissolve this in water, it rearranges, and you get a multiplicity of products. So it's very complicated. So summary, I'm not going to read this because I told this, just a final speculation. What is the active ingredients, the active species in the platinum blues? It is my belief, and not only my belief, that it may be just a transporter for the diaqua species of platinum. I have I tell you why this is the case. And I remind you, during the time I was with Barney, I crystallized this compound, the nitrato species, and we got an X-ray structure on that. I had expected this to be very toxic because the nitrates come off very quickly. But much to my surprise, it was found that this compound is very active. It is active at about 1/10 of a concentration of cisplatin. This is different from when you take cisplatin, add silver nitrate, and get a mixture, supposedly the diaqua species. But the diaqua species forms these hydroxyl-bridged species, which are terribly toxic. Now, final slide. You may not know, or some of you may know, that there is a country in the world which has this, used to have this special name, Cisplatina, and that's Uruguay. And this comes from the Rio de la Plata. It's on the cis side, adjacent to Brazil. And this is why in the old days it had this name, Cisplatina. And there they have very good chemists, I can tell you. But they get red, platinum red. And this will be my final slide. This has very, very low toxicity and is a wonderful red wine. With this, I would like to stop. Thank you for your attention.
So we have questions.
Wagner. I was in the lab at the same time that he was doing research with cells. And the question I have for you is, have there been any experiments done, maybe this will come up later, with biological molecules, either with proteins, nucleic acids, and even up to larger things with cells and so forth, to show any differences between the biological activities of the molecules that you've shown here?
You mean the model compounds I showed you? Any of the compounds. We have, in the beginning, we send out some of the compounds for testing. They were all, so to speak, active. But to be honest, industry was not really interested in these compounds because they said, we already have cisplatin and carboplatin, and they do the job. We know for sure that the bis ligand complex, so 2 uracils to cisplatin, is inactive. It doesn't do anything. But for example, Steve Lippert or Steve Hollis, who is here, he actually did this work. The 1-to-1 complexes, and that was cisdiamine platinum cytosine chloride binds to DNA vividly, and as far as I know, it also has anti-tumor activity.
Okay, because we are behind the schedule, we'll stop here and we'll leave questions to the break session.
Okay.
Thank you.
Thank you.